This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 279, recorded on April 4th, 2014. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
How's your weather out there?
It's actually been pretty nice. It's, right now it's overcast and rain's supposed to roll in. It's about 10 degrees C. But yesterday and into this morning, we had gorgeous, clear blue skies and warm by comparison to what came before.
Yeah, it's been a pretty good week. It's not winter anymore, that's for sure. And today it's gray and rainy. I was talking to someone in Minneapolis, and they say they're having a big snowstorm out there in Minnesota.
We do not want.
We do not want. Let's see what's happening in Michigan. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
Hey, Kathy.
How's the weather out there?
So we have 43 Fahrenheit, 5 degrees Celsius. It's kind of meh, foggy, kind of rainy. But I did hear that in the UP, there was going to be something like 14 or 17 inches of snow. So the UP, for those who might not know.
Isn't that pronounced UP?
Yeah, it's the Upper Peninsula. And all of us who live under the bridge are trolls.
That's so funny.
Yeah.
Well, the whole state of Michigan is sad this week, right?
Well, I guess, yeah. Michigan and Michigan State lost in basketball.
I bet Michelle is very upset, right?
Haven't seen her, but yeah, probably some glumness among the sports fans.
I get these notifications on my iPad, and the New York Times really is the only one, and they announced that both teams had lost, and I immediately thought of Michelle. I said, oh, sorry. Okay, that is it for today. It's the trio. No, not the show, but that's enough. We have 3 people. I think we had 3 last time. No, Dixon was here.
Dixon was in, yeah.
He's not here today, but he might pop in. We have one follow-up from our friend Robert. And he writes, hello, TWiV docs. I enjoyed the discussion of the zinc finger treatment paper. During the discussion, the question was raised concerning the origin of the CCR5 Delta 32 mutation. About 4 to 18% of people of European descent have at least one copy. There has been some speculation that the mutation conferred selective advantage for people with the mutation during plague epidemics of the Middle Ages in Northern Europe. Current hypothesis is that it provided some protection against smallpox throughout Europe. Also, there was a question about whether or not CCR5 deletion may be dispensable. This is most likely not the case, as a study has shown that CCR5 is important in resistance to West Nile virus infections in humans, with people carrying the Delta 32 mutation being more susceptible to infection. Weather here in Orange, California has been sunny with temperatures in the low to mid-70s. Light winds good for sailing. We did get a good shake last weekend from a 5.1 earthquake located about 30 miles inland from us, along with several aftershocks in the 4.1 range.
So CCR5, damned if you do, damned if you don't.
So these people have been trying to figure out what would have fixed this in the population. Smallpox has pretty much been discounted. I talked to Grant McFadden about that. He doesn't think that's likely. The plague, who knows? Whether it's dispensable. So the problem is that you could really only figure that out in the homozygotes, and there really aren't too many of them. So it's hard to get good information. And
so what you need to do is culture a pure population and then do a plague assay.
Yeah, there you go. Prairie dogs. So the West Nile observation has never really been... confirmed. So that's why we left it at that. Yeah. Anyway, it's an interesting question.
But yeah, it probably is useful for something.
I suppose, yeah.
Or has been.
Mm-hmm. I suppose, but I think it's hard to tease out, essentially. So
yeah, and anytime there's a skew in favor of something in a European population, people immediately think of the Black Death. This is
Right, because so many people
major
were
selective pressure that we all
out.
know about, and gee, was that it? So,
All right, that's our follow-up, and now we move on to our paper, which this was actually sent to me last Sunday by Dixon Despommier.
hey, he reads papers.
Well,
Cool.
he has this Yahoo News scan,
Okay.
and he picked up a story on The Verge, which was 2 paragraphs about this, and sent it to me. And I went and got the paper and thought it was pretty cool.
Yeah.
It is Nature Structural and Molecular Biology. The title is The Retrovirus HERV-H is a long non-coding RNA required for human embryonic stem cell identity. The authors are Lou, Sachs, Ramsey, Jacques, Gok, Burke, and how do you say NG?
I think it's
Okay. So that's what we say all the time. And this is from Singapore, Montreal, Canada.
Singapore and Montreal.
Yeah, that's it. I thought this was a really neat paper. We need a little background first. There are a couple of things here which we may have talked about, but my feeble mind has forgotten, so let's review. HERV-H. HERV-H, human endogenous retroviruses. These are remnants of old retrovirus infections, millions of years old. As everyone knows, the retroviral genome is integrated into the host genome as a DNA copy. And if it goes into the germline, it's an endogenous retrovirus. We have a bunch of them, and HERV-H is one of them. And HERV-H, it is estimated to have infected our precursors about 25 million years ago, because Homo sapiens, of course, wasn't around 25 million years ago. But you can do some kind of phylogeny to figure that out. So HERV-H is the subject of this. And the other important part is ES cells, embryonic stem cells, which I'm sure we have talked about at some point.
Yeah.
I know we talked about the induced stem cells.
Right.
So ES cells are cells derived from the inner cell mass of the blastocyst, which is a form that is implanted 4 to 5 days after fertilization. Eventually will develop into a full-fledged embryo and fetus and so forth and person. But these cells, if you take them out, you can culture them, and they're pluripotent. They can differentiate into basically all the cells in the body. Right? And these are the subject of much controversy, of course, because there are some ES cell lines out there. But when you make a new one, you destroy the blastocyst, of course. So hence the controversy about making these. Now, we talked a while ago about the fact that you can take a differentiated cell and you express, I think, 3 or 4 genes in them.
I thought it was 4, but now
it's 3 or
I'm
4.
wondering. Yeah, it's 3. It's a very small. It's a shockingly small set of genes that you express in them.
You take a differentiated, say a skin cell or something, and you express these 4 genes, and they become pluripotent embryonic stem cell. They're called iPSCs, induced pluripotent stem cells. And this is just amazing. This of course, got Shinya Yamanaka the Nobel Prize in 2012. We talked about that. Yeah. So basically, the idea is that ES cells have a very different transcriptional program. That is, the RNAs that they produce or express are very different from the RNAs produced in a skin or a muscle or a heart or a lung cell. There are a lot of them, but they're just different. And they are different because of basically transcriptional regulatory proteins. And those 3 or 4 genes that you put in, OCT4 is one of them, SOX2, KLF4, I forgot the other.
Yeah, so biologically, these are essentially the same as the cells that that were in this blastocyst that would have become a full, organism, and so they're able to differentiate into all 3 major tissue types, and they're able to differentiate into all the organs in principle.
Right. So all the proteins or the mRNAs that are expressed in ES cells, as soon as it begins to differentiate into something else, they change. All right. So it's really amazing that you can make them from 3 or 4 proteins, but that's the key, and that's very important because ES cells, stem cells, of course, can be used for a lot of medically important procedures, replacing tissues and so forth.
And besides getting around the controversy of how you generate the cells, one of the great things about the iPS cells is if you can generate them from an individual, then whatever you produce from those cells, if you produce some new tissue and you want to implant it back into that individual, they're not going to reject it because it's self.
Right.
Right. Is the fourth factor perhaps this nanog that's in the Nature paper that we read?
Could be.
I've just looked on MGI, and nanog is not the same as KLF4, so...
Might be nanog, yeah. I know OCT4 and SOX and KLF were 3, because Dixon was asking us what they all meant.
That's right.
I remember
that now, yes.
I'm sure we'll get a follow-up from... I'm sure many of our listeners know what these genes are.
Yeah, there's 3 or 4. I mean, in fact, there are... Depending on how many you use, you can get efficient or less efficient differentiation, right? But I think his original observation was 3 or 4. I mean, why don't you look it up while we're talking? The other component of this paper are long non-coding RNAs, LNC RNAs. And these are relatively recently discovered. They're defined as RNAs longer than 200 bases. And they look just like mRNAs, but they don't have a coding region. They don't make protein. And they're apparently in the human genome, there are about 35,000 of them from 10,000 different loci. And
so it looks like our ambiguity is not entirely misplaced here. There are a couple of essential. You've got to have the OCT3/4 gene from that family, and then the SOX gene family turned on, and then you've got some others that increase the efficiency, and it's like a half dozen-ish.
Yeah.
Nanog is one of them.
Which did he use in his original?
That was the next thing I was going to look up.
He has a Wikipedia page.
Yeah.
Some of these non-coding RNAs are from between genes, and some of them overlap 2 genes. Okay, and they have a variety of regulatory functions. They apparently do a lot of important things in the cell.
So Yamanaka's original paper from Cell in 2006 was 4 factors: OCT3/4, SOX2, CMYC, and KLF4.
CMYC.
CMYC, right.
CMYC, yes.
Yeah. Okay. CMYC. MYC.
So,
yes.
And they found nanog was dispensable.
Okay. Back on these long non-coding RNAs, they don't have an open reading frame, but they have to define a certain size for their cutoff for open reading frame. Do you know what that is?
I don't. I just know the size of the
whole thing overall.
I don't know. Right. If they're overlapping 2 genes, they probably have bits of each, right?
Right.
I don't know what the cutoff is because everything has little open reading frames, I guess, or can have little ones. All right. So those are the backgrounds that you need for this. And in this paper, they're looking at HERV-H. Now, there's been some data that the evolution of cells that are pluripotent, like ES cells, involves transposable elements, things that move around the genome and can contribute regulatory sequences, which would influence a transcriptional program, for example. So they decided to look into a retrovirus, which is a form of a transposable element. And they focused on HERV-H because a paper had been published, before from Jeremy Luban's lab.
We know him.
We do know him. You were here when he was here, right, Alan?
Yeah.
He's now at the University of Massachusetts. His laboratory showed that HERV-H RNAs are specifically produced in human embryonic stem cells, and they're not in a lot of other places. And they seem to be a marker for pluripotency, okay, which means that when the cell's pluripotent, HERV-H is there. When it's not there. So these guys decided to investigate HERV-H. I'm sure Jeremy is too, and he's probably not happy that
this came out.
This came out. But in the general sense, you should be happy because science is progressing, right?
Yes. Yeah, actually, when I saw this Luban paper that you linked to, I looked at it and I was looking through it, and thought, wow,
Yeah.
they came pretty darn close here.
Mm-hmm.
Yeah. So a lot of this is very interesting bioinformatics, actually. They look at data sets, and they basically looked at some sequencing data sets. They found in human embryonic stem cells 231 HERV-H insertions, okay, distinct places in the genome, which are highly expressed in embryonic stem cells. So they said, this could be important. So they used RNA interference to reduce the levels of HERV-H in embryonic stem cells. And what happens is when you knock down HERV-H, the cells start to look like fibroblasts. And they have pictures of this here. And of course, a fibroblast is a differentiated cell. It looks very different from an ES cell. So the implication is you're knocking down, you're turning off the HERV-H, and the cells are differentiating.
Right.
It's very cool. And when this happens, the markers of pluripotency like NANOG and SOX2 and OCT4, as we talked about, they go down. Right?
Mm-hmm.
Very cool.
Mm-hmm.
Yeah.
And then when you take a fibroblast and reprogram it to become an induced pluripotent stem cell, HERV-H goes up. Now, this is all an association so far, but we're going to get even better. But I don't know. I think the knockdown and the differentiation for me was just so cool.
Mm-hmm.
Yeah, that was. I'm sure it was for them too.
I mean, this is a viral RNA, right? A transcript being produced from endogenous retrovirus. And I should point out that this endogenous HERV-H does not produce particles. The open reading frame has basically been trashed by 25 million years of mutation.
Right.
So proteins are not made. It just makes a transcript, apparently. Okay. Then they start to pursue this, and they look at expression of genes nearby all these HERV-H's. All right. Now, remember, they had 231 HERV-H insertions. Of those, 128 had an LTR on either side. Now, the long terminal repeat it contains the transcriptional regulatory sequences of the retroviral genome. On the left, there's the promoter, and on the right, there would be the transcriptional terminator. So these 128 that had LTRs, those HERVs were expressed into mRNA. And furthermore, 95 of those were within 50,000 bases of a transcript, what they call it, an annotated transcript, which means that it encodes something that's known. Is that right? It encodes a protein, right?
I think
Annotated
that's right.
means it's been written down in the sequence database. And they also find that a lot of these HERV insertions are near regions of the genome that produce long non-coding RNAs, not protein-coding genes, 33%. And so that's all of that is very interesting, and suggests that these HERVs could be regulating these other genes because they're so close to them. Now, remember we talked about this experiment where they knocked down HERV transcripts with RNA interference. They find that most of the transcripts that they're looking at here, they're all depleted by this knockdown. And in addition, the long non-coding RNAs and the protein-coding RNAs, which are near the HERV-H's, which I just mentioned, those are also downregulated too when you knock down the HERV-H itself.
Right.
So you knock down HERV-H, and then all of these close by genes and long non-coding RNAs are knocked down as well. So that suggests that the HERV-H is somehow influencing the expression of these neighboring genes in the long non-coding RNAs.
Now, just to toss a little bit of a wrench in this, anytime you do these sorts of experiments with inhibiting the genes, there's leakage, there are off-target effects, this is not a perfectly precise elimination of the activity of the HERV-H, but they do some controls to try and narrow that down, and everything does kind of line up nicely in this, but it's not It's not an either-or situation.
Right. Now, the next thing they said is what does the HERV-H do? Is it an enhancer? An enhancer, of course, is a sequence that can influence the production of transcripts at a distance. And since, they found these HERV-H's, not right next to but close to genes, then being an enhancer could be one way that it might influence their expression. So they molecularly clone the LTRs of the HERV-H in the genome of these ES cells, and they put a reporter protein next to them, and they show that, in fact, they have enhancer activity that's specific for human embryonic stem cells. And if you knock down OCT4, right, one of those pluripotency markers, the enhancer activity of the HERV LTR goes down. So this is getting intriguing now.
Yeah.
You're seeing the LTR seems to contain an enhancer. The genome influences neighboring genes, and OCT4 is apparently needed for this LTR to act as an enhancer.
Right.
Now, this HERV-H they find is in the nucleus of the embryonic stem cells. And they say since, links are known to be enriched in nuclei, this suggests that the actual HERV-H may be a long non-coding RNA itself because it doesn't encode any protein, remember. So not only is it near long non-coding RNAs and can influence their expression, but it itself appears to be a long non-coding RNA.
Right.
So the idea is arising that maybe this viral long non-coding RNA is some kind of a scaffold, and it's binding other proteins and recruiting them. So they do a series of very interesting assays where they look at what proteins bind the HERV-H transcript or genome, whatever you want to call it. And they find a number of transcriptional regulatory proteins, which include coactivators like CBP and P300. These are proteins that they don't bind DNA directly, but they interact with proteins that do bind DNA. HERV-H also binds OCT4 and a number of other transcriptional regulatory proteins. So this viral RNA is binding proteins that are important for the pluripotent status of these human embryonic stem cells. An experiment they mentioned here, which I thought was coming up before, you knock down HERV-H. Again, the LTR, the enhancer ability of the LTR is also down. And you also reduce genes that are near HERV-H when you knock down HERV-H itself. Okay. So this is basically, that's the data basically. So the model is HERV-H RNA, which is a long non-coding RNA because it doesn't encode protein, interacts with transcriptional regulatory proteins to establish a enhancer at the LTR of HERV-H, which then it helps to turn on neighboring genes and long non-coding RNAs that are important for stem cell identity. And then you knock those down by knocking down HERV-H. They're no longer stem cells. They differentiate into other kinds of cells.
Mm-hmm. Now, one of the things that it's a neat model, and I think the data are really, really cool. But as I was reading this, one of the things that kept running through my head was, how do you end up evolving a system like this this recently? Because if this is something that came in, what did we say, 25 million years ago?
Yeah.
There's been multicellular life a lot longer than that. And all animals pretty much have to deal with this stem cell production issue, and you have to have a blastocyst. And so how do you end up with a relatively recent retrovirus coming in and becoming part of this process? And I have some thoughts, but I'd like to hear yours too.
My thought is that so this is a primate-specific endogenous retrovirus. It's not in fish or birds or whatever. So my view is that we probably had a way of becoming of being pluripotent before, and then we got infected, and at some point this turned out to be better.
Right.
And so it was selected for. I guess that's one explanation. What do you think, Kathy?
Well, that seems reasonable because it's analogous to the situation with retroviruses and the placenta.
Yeah,
right. Because
Right.
retroviruses don't explain all of the placental, if I understand that correctly, all the placental evolution. So it doesn't have to explain all of life that develops from stem cells.
Right.
Right.
But
Yeah, that's kind of the same line I was thinking along. There was a system to do this, and this system came in and got dragged into the process as part of its stabilization in the genome.
Would be interesting to look in non-primates.
I'd love
Right.
to see. I'm sure probably these folks or somebody else, maybe Jeremy's lab, are looking maybe in mice.
Yeah, and see if
Be easy.
there's another ERV that's doing it, or if they're not involved at all. That could be.
And it
So-
might explain why iPS cells production is inconsistent from species to species. You have to do, you know,
What were you going to say, Kathy?
I was just going to say, in mice, then the ERVs are called MERVs, mouse endogenous retroviruses.
so.
Yeah, that would be Merv.
Okay,
You have if Jixon were here, he'd have a Merv joke. But
probably.
Alan should have a Merv joke.
Merv Griffin, maybe.
Yeah, Merv Griffin.
Yeah.
So basically,
before my time.
so the cool thing is when you put these four genes, these four transcription protein genes into cells, into differentiated cells, then you're basically turning on HERV-H, and then HERV-H is doing things that keep the cells pluripotent. It's just amazing.
Yeah.
And without it, it won't work. They will not. They will not stay pluripotent if you take away HERV-H. They did that experiment here. So presumably, well, I guess you couldn't develop, you couldn't have an embryo. You wouldn't have pluripotent stem cells at all.
Right.
I just think it's cool that there are lots of things to be done, clearly, but it's very cool that a virus could provide this function.
And it ensures HERV-H's preservation in our genome.
It sure does, unless something else comes along.
Unless something else comes along that does the job as well. Yeah.
But there are a lot of copies of it, apparently, right? And they're all next to areas that are probably important for being pluripotent. So we are viral,
Yes, we are.
Right.
right? There's just no doubt. We're viral, we're bacterial. I mean, without this infection, who knows what we would be like? Maybe we would never evolve into hunter-gatherers.
Maybe we'd be ferrets.
Oh my gosh. Then the H5N1 transmission experiment
Would wipe us out.
Oh boy. Well, anyway, that's a brief communication, but really, really cool data. Unfortunately it's behind a paywall, so you won't be able to get it, right?
Right.
Yeah.
But
I
you
just
can-
had one tiny little quibble with the title. Maybe it doesn't bother you guys, but it's just the retrovirus HERV-H is a long non-coding RNA required for human
Yeah, that's
embryonic stem cell identity.
Right.
The is part threw me.
Yeah, the retrovirus is not the genome, right?
Right.
It should be retroviral HERV-H genome or something like that.
Also,
Yeah.
is HERV-H still a retrovirus?
Well, it depends how you define life, Alan. I don't know. It's got 2 LTRs, and probably you could probably find remnants of gag Paul in there.
Yeah, it
sequences.
certainly was a retrovirus.
Yeah, I mean it's a transposable element now, right?
Yeah,
I think
right.
they call them retrotransposons, really.
Right.
So to say virus is a bit, yeah. I
But
agree
then
with
part
that.
of its name is endogenous retrovirus. So maybe
Yes.
that's not. I think I would have just changed the is to encodes. The retrovirus HERV-H encodes.
Yeah, that would be
a long
better.
non-coding
Yeah.
RNA.
Come on, nature.
Hey,
What's with the editors there? Why don't they fix? They don't change titles, right, Alan?
I don't know. They would probably kick back a title if it.
Well, if the reviewer said something about it,
Yes.
So.
Well, and the editors at Nature do read the paper first. They triage the papers, decide what editors to send them to. So if there's something in the title that really sticks out, I guess they probably flag it.
My copy here, I'm on the website. It says access provided by Yale University Library. I'm not at Yale University.
Huh.
Neither are the authors.
That's weird. The Nature website's been having problems for the last day or so.
Yeah, I tried. Normally, for Nature papers, I get them through the press website at Nature, and that just is frozen. So I don't know what's going on there.
Now, I have to ask, these are very beautiful images here, these mandalas. Did you put those in there, Kathy?
Right. That was relating to the other paper, the Jeremy Luban paper.
Oh, okay.
They had the mandalas that were how they showed the proximity of the HERV-H provirus to specific genes in the genome, and they used mandalas. So I thought, what is that word? And the first thing I found were these beautiful mandalas.
That's nice. That's very good. And I must say, I like Nanog. It sounds, I don't know. It
sounds like Robin Williams' Mork and Mindy, kind of
Nanog.
It's a neat name.
Yeah.
Or it could be from Tolkien.
Yeah, it could be too. All right, let's do some email. Alan, can you take that first one?
Sure. Bill writes, Vincent et al. I read Slashdot daily. Today they had this article about PLOS, which you might like to read. He gives a link. Here's the front door portal for Slashdot, which is slashdot.org. And there's a wiki page about it as well. And then Bill goes on. I made this comment as Horizon, my nom de plume on Slashdot. A lot of people ignore the collateral functions of the peer review system administered by the publisher. So this is talking about the backstory on this is PLOS is now going to require public access to the data behind a paper.
Right, instantly and all of the data.
Instantly and all of the data, and this has been somewhat controversial on all sides. But anyway, Bill's comment on this that he posted on Slashdot, a lot of people ignore the collateral functions of the peer review system administered by the publisher. The publication must be read by someone who knows the subject passably well. If his first pass finds it acceptable, he must then select from a number of true experts in these matters, the peers or equals to the writer of the paper. He works for a living as a competent editor for that area of research. The peers he chooses are sent a copy of the paper to review and criticize if it's not acceptable. If it's not acceptable, the comments are passed back to the author for him to respond. After his responses to fix the flaws, it goes back to the panel and so on until rejected or published. The review mechanism is needed to avoid total BS being published. I would just add that it doesn't automatically prevent total BS from being published, but yes, that's its purpose. The publishers have created this niche and profit by it. Some say excessively, and I agree. So some way must be found to pay for these. Page fees are the initial solution. The author pays a fee, and this is spread among the experts involved. As for an archive, in the USA, the Library of Congress can do this as long as a proper indexing method is used so that the paper does not become a needle in a haystack. It should be Google indexed. Perhaps Google will fund this via ads because all the biological supply houses will place biological ads, and the same with all the other disciplines. In fact, this could become a gold mine for Google and at the same time serve PLOS and the research community very well. Large databases of terabytes of particle data would not be stored. The publisher would grant access to those who wanted to download it. A precious few will want terabytes of particle data. So why not someone who has a pipeline to Google give them a whistle? They might leap at the chance. It's a natural fit.
Kathy, what do you think about this putting your data up there?
Well, I just started to read a few of the comments on that site, and the first one or ones was from physics, and they said there's petabytes of data, and if you don't know how to deal with the data, it's going to be meaningless, and it's going to be a fair amount of work and expense to put the petabytes of data in a way that can be accessible. Yeah. So, for something like that, I can see the point.
For us, though, we don't have that much, right?
We don't have petabytes, maybe not even terabytes. So yeah.
So if you had, let's say you had a nice figure of a Western blot where you've cut out the one band and you show it across different conditions, for the data, would they want a scan of the original?
Sure.
The X-ray? Yeah.
Yeah.
So that's going to add some work for us, right?
Yeah, you'd have to scan in all the films and somehow label them. I mean, as it is now, supplemental data can be sometimes totally useless because if it's not peer reviewed, the authors in the journal don't do much with it, and it's meaningless, because you don't really know what they did.
Yeah.
So this could only make that worse.
So for a graph, you'd have to give them the numbers that you use to generate the graph, right?
Mm-hmm.
Just to put this in perspective, too, demanding to see the raw data has been for years a very, very common tactic for harassing scientists who have published research that corporate interests find inconvenient.
Mm-hmm.
Mm-hmm.
And it turns out that when somebody, under certain circumstances, they can demand your data, and you have to then dig through all of your lab notebooks. And Vincent, I mean, you just commented you don't have that much data, but I know that if you walk into your lab and you look at the shelves and shelves and shelves of laboratory notebooks,
yeah,
you quickly realize if somebody demanded to see all those data, how are you going to do that?
Yeah.
Give them the key to the lab.
And do you do you include all of the runs of the experiment that didn't work?
Oh, that's a good question. Yeah, I wonder.
Because, I mean, that's like 80% of my graduate notebooks are...
Hey! Yeah, it's...
repeating this PCR reaction for the 39th time.
Well, someone asked that over at PLOS, and they said, we're not saying what you should upload. You decide. Just put data there, So they don't want to tell you. So now
they don't even care if it's all the data.
I think if who's going to go through and check?
Well, that's the thing, is that they made this... I think this was maybe a little poorly handled by PLOS. They made this blanket statement. You're supposed to put all your data online and make it immediately accessible. And then in the subsequent screaming about this, and in the case of people who are working on whole genome sequence or, transcriptional profiling, some of these other types of experiments, they are biologists with petabytes of data.
Yeah,
true.
And they're looking at this and saying, you want what? Where?
Yeah.
That's the other thing. First, that they cannot possibly check because it's too much work. They don't have the staff. And secondly, where? If I put my blots on a Google Docs document and make it accessible, is that enough? Or and then they don't tell you. They don't give you any guidance. They make a few suggestions. But
in the subsequent flare-up, there's been a lot of either backpedaling or just an acknowledgement that they hadn't really thought this through.
Nice.
Where the folks at PLOS are now saying, well, you can interpret what all and immediately mean on your own. So maybe you can say that what you published in the ordinary paper was all of the data and that's immediate. I don't know. But what they're trying to do is make science more open, and I applaud that notion, but I think more thought needs to go into some of the problems of doing that.
Yeah, some of which have come up just in this brief discussion, right?
Yes.
Mm-hmm.
It's an interesting issue, though.
Yep.
All right, Kathy, can you take the next one?
Sure. Pavitra writes, Hello, TWiV. This is in response to a letter you read on TWiV 273, where Bill asked about a way to make negative results available. I just wanted to bring to your attention a couple of journals that might be relevant. There are probably more, but here's one in biomedicine, and the link to that is to the Journal of Negative Results in Biomedicine, and one for ecology and evolutionary biology, and that's just called Journal of Negative Results, and the subtitle is Ecology and Evolutionary Biology. I didn't know those journals existed. I also remember reading about a journal of serendipitous and unexpected results, but it appears that their website has been rather unexpectedly taken over by a Japanese company or spam bot.
It's all Japanese or whatever.
And it looks kind of like a generic type of page, so there's no telling what this is.
So you can only publish negative results at these journals?
Well, the mission statement for Journal of Negative Results, which is the ecology and evolutionary one, says, publish peer-reviewed sound scientific work that is scientifically rigorous but does not rely upon arbitrary significance thresholds to support conclusions. So
it's funny. Volume 10, number 1, table of contents. There's one article.
Yeah.
It's kind of lonely there, isn't it?
Yeah.
Well, is their impact factor negative?
Yeah.
It's interesting. So we actually have 1, 2, 3, 4 so letters on negative data. I put them all together here.
Yeah. And this actually kind of ties in with the previous letter.
It really does, yeah.
Because it's also about data that are not normally out there.
Yep.
And in this case, as people have been talking about, and as we've talked about here on TWiV, it's a problem that you've got all this stuff sitting in file drawers that could maybe save somebody from doing the same experiment, getting a negative result.
Yeah, so much of it, though. I'm not sure Google is going to be interested.
Right.
I'm not sure they can monetize it.
And this sort of thing is going to get very few hits.
Oh, yeah.
Because nobody's going to cite it. They're going to look at it, say, "Oh, well, we don't want to do that experiment." And then when you do publish, you're not going to cite all the experiments you didn't do.
The next email about negative results is from Raihan, who writes, "Hey, fellas, I came across this paper from PLOS ONE showing negative results and rushed to show it to you guys with hopes to share the great news. You can publish negative results." The group started with the hypothesis harbor porpoises are a reservoir for influenza B viruses. And after serological analysis of 79 porpoises, no antibodies were found specific to influenza B or A viruses. Therefore, they concluded that their hypothesis was wrong. You know, just to interrupt Raihan, that might not be correct. It may be that in harbor porpoises, antibodies decline very quickly and you can't detect them after infection.
Or that those 79 porpoises didn't get it.
Yeah. Mm-hmm. And he continues, "I know this paper might not be significant for a TWiV discussion, considering the other cool stuff you guys discussed, but you may remember from a letter I wrote in a many TWiVs ago that influenza B virus is my baby, with 2 capital Bs. And from now on, all the Bs are capitalized. I can't help but rush at any publication about it. Hope this email gets read. Wishing you all the best for TWiV. Best regards and best wishes." That's great. I like that.
Yeah.
I do remember his original email.
Mm-hmm.
So, yeah, this is a paper in PLOS ONE, and, yeah, no serological evidence.
You have a good suggestion here, Kathy.
Yeah, because he likes bees, he could work on influenza B and the blood-brain barrier, or he could work on B lymphocytes. Mm-hmm. That way he could capitalize more bees in his research.
I wonder if we could make a title in all bees. Okay, that brings us back to Alan.
Joseph writes, Greetings TWiV docs. I'm writing to offer my 2 cents on the negative data discussion stemming from an email read on TWiV 273, as a substantial portion of my PhD thesis was negative data. At the time, quite a number of good papers were published demonstrating that the macroautophagy pathway contributed significantly to the generation of peptide epitopes during an antiviral T-cell response. Because we were very interested in the pathways of antigen processing, we wanted to see if this pathway was important during influenza virus infections. Despite our best efforts, we could not find any significant contribution of macroautophagy to T-cell activation. Sure, we could force it to happen with artificial constructs and controls, thereby validating our detection methods, but the data indicated that this pathway did not contribute to T-cell activation, at least at our level of detection anyway. This was difficult to get published, but after many revisions and rebuttals, it was accepted at JVI. Looking back on this only after a few years away, I appreciate those revisions and rebuttals much more because the paper was much stronger at the end. Dr. Condit is dead on about the purpose of meetings and communication. I always found 1 or 2 other people at meetings that were making the same observations as us, but also could not or would not publish their data. I suspect that the phrase negative data leads many to conclude that the data are not important. As you all discussed, I think this couldn't be further from the truth. Negative data can indicate that our procedures or techniques were wrong, that the technologies we used were inappropriate, or that our hypothesis needs revising. That's really helpful information. Obviously, not all of it is publishable, but helpful nonetheless. Maybe this inappropriate interpretation of negative data, particularly in the general public, is a result of how science and the scientific method is taught in schools. In the introductory biology classes for non-majors that I teach at a local university, I'm always careful to explain the scientific method in detail, as this description in most survey text is incomplete and more or less depicts a negative result as the end of a line of questioning. 2 more quick points on this subject. First, what may be negative data today might be positive data tomorrow with advances in technology and the development of more sensitive techniques and procedures. Second, at least in my experience, there are also positive results that do not get published for a variety of reasons. That is very true. I have a pick of the week, if I may, a website from UC Museum of Paleontology, University of California at Berkeley, called Understanding Science. It has a really nice overview of how the scientific method, and therefore science itself, works, from screening the literature to collaboration with other scientists, and gives a link to their main page and the scientific method page. P.S. The weather in lovely Doylestown, Pennsylvania is 24 Fahrenheit, minus 4 C, with a light snow falling.
Nice website.
Mm-hmm.
I've been to a museum at Berkeley. I guess it's the same one. They have lots of skeletons of dinosaurs in the atrium. They have a nice T-Rex and a pterodactyl hanging down. It's pretty neat.
This is nice.
Mm-hmm.
So is he being sarcastic that Doylestown is lovely?
Oh, I don't know.
Yeah. So this is important. Not negative. It has a bad connotation, obviously, but
It has a negative connotation.
it does.
Yes.
And sometimes you have to. You prove your hypothesis is wrong, and that's it. But it shouldn't be. Maybe there should be another word. Null data. It's no good, huh? Well, it's there. We're not going to change it.
Right.
Anyway, thank you, Joseph. That brings us to Kathy.
John writes, Dear Triumvirate?
Nice.
Yeah, so I guess that's TWiV, TWiP, and
Triumvirate.
TWIM. Okay. I've wanted to write to you guys for a long time. Your podcast started early in my time as a PhD student at Dr. Racaniello's alma mater, and I've listened to it pretty regularly ever since. I don't know why I've not lent fingers to keyboard until now, but I guess I haven't really felt like I had much worth saying. At the moment, the weather here on the east side of Manhattan is 1 degree below Celsius. As it is Friday morning, that probably matches the conditions for the recording of TWiV number 275. What little snow remains is a sad reflection of its former self, embedded thoroughly with the schmutzstecke of New York life. A while back, a listener sent in a piece I'd written, Why You Don't Actually Effing Love Science, which was brought by Mashable, and you guys briefly discussed it, but that's not what I'm writing to discuss. What I wanted to write about this time is a theme that comes up from time to time during the course of the show, the publishing of unpublishable data. I mean negative results that are difficult to reproduce, etc. Recently, I remember someone wrote to your show about how publishing data like this would prevent wasteful investigations. I don't really think we should prevent those investigations. Just because someone has gone looking for a finding in the past and failed doesn't necessarily mean that the case is closed. Negative data is always weaker than positive data, which is exactly why it's so hard to publish. There's no telling why an experiment might show nothing, and it's not always because the hypothesis was wrong. Sometimes it was just a bad month for that postdoc's experiments. That said, I think that it pays to have the ability to look up what others have done in the past and use it to inform future investigations. Knowing what methods failed in the past and being able to update them presents a starting point for anyone else who might want to try their luck. Of course, as TWiV has discussed in the past, peer-reviewed journals are reluctant to publish negative data because it's difficult to be sure that the negative result is due to reality rather than experimental artifact. Negative data has a lot of trouble in peer review. My question and solution are therefore, why bother with peer review of negative data? The point of peer review is to ensure that what we know about science fits with the understanding of fellow scientific experts. I think that mostly is important for positive data. If you base an experiment on incorrect positive data, you might spend years chasing your tail when it doesn't work. On the other hand, an experiment based on incorrect negative data, when it reveals the true positive information, will just lead to a novel result. The problems that peer review is supposed to solve don't necessarily apply to negative findings. Why not just dispense with peer review of negative data and have an online database of informal research reports on ideas that didn't work? It seems to me that this could be a valuable resource for investigators, study sections, and others. Everyone would treat it like a conference abstract book, out in the open, but not necessarily sound in its foundations, a sort of guiding light for investigations toward peer-reviewed publications. As it is, this kind of informal information is shared among colleagues within a given institution or among people who have made connections with each other over the years. This rumor mill isn't efficient, though, and while it may seem to be a sort of tactical advantage to have insider knowledge that others lack, this advantage comes at others' expense. Too many people waste their efforts because there was no mechanism for them to learn what you know. That in turn hurts the overall scientific community. The thing is, I have no idea how someone would set up such a database. I don't know how it could be funded, who would do the work of keeping it running, or how it would be publicized. Fortunately, you folks have done all these tasks with the TWiV podcasts, so I thought I would run the idea by you. So what do you think? Thanks for your time and for TWiV. John.
All right, so I think he has a good point here earlier on, which is not really his question, but I wanted to highlight it, that, and I thought of this earlier, just because someone gets a negative result doesn't mean no one else should try it, right? In fact, sometimes others can get things to work or whatever. So, this all comes down to the idea or the question whether it's really important or not to push the field forward. People will hammer at it until they're satisfied that it's answered. So I agree that you don't want to prevent, and I don't know that, I mean, if you were thinking of doing an experiment and there were a database of negative results, would you comb it for similar experiments first? I don't think so. I think most people would just do it.
Well, I'm not sure that this is actually necessarily a problem.
What is that?
That people avoiding doing an experiment because somebody else published that it didn't work.
Yeah, no, I agree.
First of all, I think the concept behind most of these efforts to publish negative data, the reason you would have peer review is to make sure that it's not just that somebody's PCR reaction didn't work, that they actually have, that they've actually documented and done, the proper controls and proven that the assay works, and they really nail down that this experiment did not work and it wasn't something that the postdoc was having a bad week. So it would have a little more heft to it than just, oh, gee, I couldn't get that to work. And at the same time, I think most people in science are fundamentally, not really, we're not generally the kind of folks who just go along with what somebody else said. Would you agree?
Yeah, absolutely.
Yeah.
Absolutely. If I had an idea and then I looked in a database and someone had not gotten it to work, I would say, hey, let's try it anyway.
Yeah.
And then Alan
exactly.
would say, I don't really want to do it. And then you have to have a little argument. And then,
well, no. If you told me that somebody else couldn't get it to work, that would be motivation to go make it work. Like, hey, I'll show them. And I'm also kind of wondering what's going to happen if these negative results journals get off the ground and eventually there's some negative results paper that it's proven that the data are just flat wrong. Are they going to have to retract the negative paper?
No, I don't think so.
And if so,
John,
would that count as a new publication?
John says it's not going to be peer reviewed. So.
It's not going to be peer reviewed, so it'd be sort of like Negive instead of archive.
So the problem is that I can imagine this being just a collection of junk, right? People just put there, or maybe they wouldn't bother because it takes time to even post junk. You have to have some mechanism for parsing it, right? For making sure they're legitimate laboratories submitting data to it. But it's a huge undertaking to maintain. It costs money for the servers. I think it will probably happen someday. Someone will try it, John. Someone will do the experiment, and I don't know what will happen. But I just think it could get very unwieldy, right?
And it's a question of who's motivated to do this.
Yeah.
So here at TWiV, we don't really have huge databases. We just have show notes for 3 different shows, which is not really hard.
We don't even have transcripts.
Yeah, it's not hard at all.
We do for some. But.
So we are certainly not the people to do this. And but I don't know, throw it out.
We do produce gigabytes of data, but it's mostly like Dixon's breathing sounds.
Yeah, every hour is like 500 megabytes of sound for each of us.
Wow.
500 megs each, and if we go to 2 hours, it's approaching a gigabyte. Yeah, I have a lot of hard drives storing all the old episodes.
Yeah.
I don't know what will happen to them, but I figured I should keep them all. They're there. I have I'm on the 6th 2 gigabyte hard drive of archived TWiVs.
Wow.
And I keep a backup of each one, so I have a duplicate.
Well, but data storage is cheap.
Yeah, it's fine.
It's if you actually want to, if we actually had, that much real solid data that somebody wanted to analyze in detail, and it wasn't just recordings of voices, then the analysis is the hard part.
Do you think you could? I mean, this needs an IT person. We have a bunch listening. Anybody's motivated. Do you think this could be funded by like a Kickstarter or something? I don't know if people would want to invest in
an archive of negative results.
Yeah. Except for scientists. But anyway, if anyone out there has ideas, I think it's a good idea, John. I don't know how it would be implemented, if it would be supportable, and so forth. Certainly, I looked at, so on the PLOS discussion we had earlier, PLOS has a few suggestions for places online to store your data, and you have to pay to put it there. So would you pay for storing negative data?
Yeah. Would your grant pay for that? Would you want your grant to pay for that?
Would you want your grant? Yeah, because you can choose to pay for it.
Yeah.
If you have a choice between that and other things, I don't think so. Interesting stuff, though. Thank you, John.
I have two related things. I found an unofficial impact factor for Journal of Negative Results in Biomedicine, 1.15.
Mm-hmm.
And then it triggered something when you mentioned that we don't have transcripts. My former student, Nikki, pointed out that there's software called Dragon Dictate, and that might be a way for somebody to take a podcast and quickly get something that then they could revise using this Dragon Dictate software.
Right.
So
I've thought about that, yeah. I haven't. The problem with Dragon Dictate is that it gets used to your voice and it learns. Mm-hmm. But we have 4 or 5 voices, and I don't know how that would work.
Maybe it could learn 4 or 5. Huh?
And I think it's designed mostly for actually dictating text, period. So, it's accustomed to inserting the punctuation along the way, period.
That's where people could,
so
as I say, you quickly get a rough draft, and then you could
you'd have to manually go through and...
I'm sure that this will be very easy one day. Google or someone will do it. In fact, we had a listener, Steve, I think, who was transcribing a while ago, and he was experimenting with different ways of getting first drafts like that. So it'll eventually be there. I mean, I think eventually you'll also have real-time translation into whatever language you want, right? So people will listen to a TWiV, and it'll be translated into Farsi.
Mm-hmm. Yeah.
All right, the next one is from Michael. Dear Vincent and colleagues, I am a regular listener of TWiV and know that you visited my institute, the Blood Sciences Research Institute in San Francisco, previously to discuss my colleague Eric Delwart's work on viral metagenomics. Of note, my colleagues at BSRI, particularly Graham Simmons and I, were quite active in refuting the alleged discovery and clinical associations of XMRV, authoring several papers in Science and other journals, including the lead and senior author of the science paper from the Blood Safety Working Group that was linked to the Science editorial decision to retract the Lombardi et al. Paper. And I do remember that, Michael.
Yep.
Absolutely. We talked about it, right?
Yes.
I'm writing in reference to the brief discussion during the preamble of session 273 on whether successful vaccines prevent infection, i.e. are sterilizing, precluding viral entry, and evidence of virus in blood or anamnestic adaptive immune responses, or allow viral inoculation and local replication, but block or blunt dissemination of viral infections, yet are highly effective at preventing chronic infection and disease. I think data on efficacy of HBV, hepatitis B virus vaccination, is illustrative of these issues. Although HBV vaccines have proven extremely effective at preventing chronic hepatitis B surface antigen positive carriers and consequent clinical hepatitis, cirrhosis, and liver cancer, long-term follow-up studies of vaccinated populations have demonstrated modest rates of anti-hepatitis B core seroconversion as vaccinated children reach ages when they experience sexual or parenteral exposures to HPV, indicating that HPV infections have occurred in successfully vaccinated recipients, albeit at reduced rates relative to historical controls. Several papers documenting the clinical efficacy but low rates of anti-HBCSC are attached. Relevant to my area of focus and expertise, international blood safety and efficacy with broader interest in virology, emerging infectious diseases, and transfusion and transplantation medicine, We have detected HBV vaccine breakthrough infections in blood and plasma donors screened using highly sensitive nucleic acid amplification technology. Several papers, including a seminal paper in New England Journal of Medicine describing such cases now observed in blood screening programs worldwide, are attached. Mechanistically, our paper, published in JID, Journal of Infectious Diseases, documents the blunted viremia which was associated with early engagement of the innate immune responses and anamnestic anti-HB surface, SC. I don't know what SC is.
Yeah.
I should look. I guess it's in one of these papers. SC. Okay, anti-HBC SC. Now I've lost my place.
Penultimate paragraph.
There we go. That we were able to characterize based on study of longitudinal samples from donors with vaccine breakthrough infections, evidenced by very low-level HBV DNA in the presence of vaccine-induced anti-HBs, but absent of HBs antigen in anti-HBC. We believe these data demonstrate mechanisms underlying vaccine efficacy in the absence of sterilizing immunity. As we note in the conclusion of the paper, further studies of viral control and immune response in the context of pre-existing vaccination are needed to fully understand mechanisms underlying vaccine breakthrough infections, such as incomplete seroconversion, waning anti-HBs, or prevalence of diverse HBV genotypes. Finally, studies elucidating consequences of vaccine breakthrough infection should be conducted to determine if there is persistence of HBV after breakthrough infection, and if these individuals are susceptible to reactivation in the context of immunosuppression, as in classically resolved HBV infections. Finally, I've attached an excellent commentary in Nature Medicine on lessons from vaccine history that makes the point of the need to understand the mechanisms underlying clinical efficacy of established vaccines as we struggle to advance vaccines for HIV and other viral and non-viral pathogens. So that's interesting. Yeah, so the answer is that there is some infection in immunized people, right? It's not sterilizing.
Right.
I think the SC might just be shorthand for seroconversion.
Could be.
Maybe.
Seemed to make sense most places where I read it. Okay.
Mike is the director of the Blood Systems Research Institute. I don't know, the director. He is Eric Delwart's boss. Mm-hmm. Thank you for that, Michael. It's very useful. And maybe there aren't so many systems where we have this kind of data, right, which would give an answer to that interesting question. All right. To a couple more. Back to Alan.
Okay. I can do the next two because they're short.
Mm-hmm.
Robin writes, MERS virus in dromedary snot has been around long and a lot. Human may end up dead, but fortunately don't spread so long as no mutants get hot.
I guess you got inspired.
Yes. Mm-hmm. And then Martin, who we've heard from before, writes, why didn't you mention this? And quotes ABC News, all of the students who were tentatively diagnosed with mumps had been vaccinated. Vaccinations do not offer 100% protection, and this is regarding the mumps outbreak at Fordham University, which happened early March, I guess. Mm-hmm. And links to this Natural News Wackaloon site, this website, Natural News, which riffs on this apparent discovery that bunch of these kids who'd gotten mumps at Fordham had in fact been vaccinated against mumps. And interesting you should bring this up, Martin, because this is in fact something that I believe we've said repeatedly here on TWiV, and I believe is said repeatedly by everybody who advocates vaccinations. Because they do not offer 100% protection, we need something called herd immunity, which is everybody gets vaccinated, and then there aren't enough susceptibles in the population to introduce the infection in the first place, so you prevent these types of outbreaks. That's why we need 100% vaccination coverage, particularly in a vaccine that doesn't have perfect efficacy, which would pretty much be all vaccines. But the vaccine is still the best way to protect yourself from mumps.
So this headline that all the students had been immunized is just wrong.
Yeah, that too.
It's wrong because some of them do happen to be immunized, but most of them are not. And it's the same situation with the current measles outbreak here in the city, which is up to 20. A few of them had been immunized, but most of them are not.
And of the ones who've been immunized, you find out that a number of them got 1 shot or 2 shots. They didn't get the full series. And even if you do get the full series, these vaccines are like 98, 99% efficacy. And that's not perfect.
Right.
Everybody involved with vaccines, it freely admits and in fact explains that they are not perfect, which is exactly why we need people to all get them, so that you don't have a large enough. You will always have some susceptible population, but you want that population spread out enough that it won't be able to sustain transmission.
Yep.
So, yeah, nothing new here.
All right. Should we do a couple more? Or do you want to stop, guys?
Yeah.
More?
Either way. Yeah.
All right, so this next one, I can't reach the nature link, unfortunately.
Oh, I got it. It took a while. I just had to be patient.
Okay.
Yeah.
So you want to read it, Kathy?
Sure. Vincent et al. This is from Dave. Have you seen this paper? And he links to a Nature paper. If confirmed, this would be huge in the field of pulmonology. Idiopathic pulmonary fibrosis is a devastating disease affecting about 1 in 100,000 people. It progressively destroys the lungs and has a median survival of 2 years. There is no effective treatment or any real insight into what causes it. The hypothesis advanced by this paper has some biological plausibility. Unfortunately, I've seen nothing further published on this, so I'm concerned that other groups are having difficulty replicating the findings. It's reminiscent of XMRV. But why I really wrote is to let you know that I've now caught up and listened to every TWiV. TWiV is next, and it's currently minus 26.6 Celsius, minus 16 Fahrenheit in central Minnesota. That was a while ago, I believe, so maybe Dave's even caught up with TWiV.
Yeah.
So this is a paper from Modern Pathology, and the title is Idiopathic Pulmonary Fibrosis is Strongly Associated with Productive Infection by Herpes virus Saimiri. And so evidently this is in the Nature family of journals, but it's called Modern Pathology. And so just kind of quickly looking through the abstract, they find herpes virus Saimiri DNA in generating epithelial cells of 21 out of 21 idiopathic pulmonary fibrosis cases using 4 separate probe sets. And they evidently have controls. So the data are consistent with idiopathic pulmonary fibrosis representing herpes virus Saimiri-induced pulmonary fibrosis. So I think the listener's evaluation is right. It doesn't necessarily indicate a causality. We know lots of cases where people had to work for many, many years to show causality. Zurhausen and HPV and cervical cancer comes to mind for me. But this might be something that could be pursued further.
Do they have controls?
So it says they have controls. Those they have PCR controls, but do they have
negative controls?
non-IPF cases?
Yeah.
And I haven't had time to look at that. Sorry.
It says age-matched controls with pulmonary fibrosis of known etiology.
Okay.
So I guess there's known and unknown etiology.
Right.
To quote Donald Rumsfeld.
Right. Yes. Right. But it's true that idiopathic pulmonary fibrosis is a big deal. And, we know that idiopathic means it's just a fancy doctor way of saying we don't know why. We don't know what causes it. So
this is a virus of squirrels.
Saimiri?
Squirrel monkeys. Sorry, not squirrels.
Yeah,
squirrel monkeys.
That was a simian. Yeah.
And so how does it get into people?
Eating bushmeat. That would be the answer I would give or my students would give on the exam.
But these are people in New York City.
Well,
from the files
which doesn't exclude that, but yeah, it's probably not.
It doesn't?
Yeah, the place down on 14th Street, they deliver.
Meat market. This is, I don't know if you remember Alan Jerry Nuovo. Do you remember him? He was with Saul for many years. He's the senior author on this paper. And he, well, I will ask Saul about this. He's not here today, but he would have an opinion, I'm sure.
Oh, yes.
Okay, that's interesting. And it remains to be confirmed, of course. This is November 2013, so it's relatively new.
Yeah.
All right. Thank you, Dave. And one more from Anne, who writes, "Hello, TWiV team. First, many thanks for all of your efforts with the podcasts on virology, microbiology, and parasitism. I was delighted to find your podcast last year, and while I am nowhere near being caught up on the virology podcasts, I look forward to listening to them when I can. My college-age daughter recently experienced an episode of Epstein-Barr virus, which results in glandular fever mononucleosis. She was diagnosed by her school's clinic, and I encouraged her to have them confirm through a blood test that was what indeed was the cause of her illness. Very common to obtain in college. And it's called the kissing disease. I heard a talk once at a meeting where they monitored the seroconversion of freshmen, and the only ones who didn't seroconvert were those who claimed in questionnaires not to have kissed anyone. While she went through about 2 to 3 week period of chills, fever, and great fatigue, she recovered without having to miss a lot of school. Her class load at school is lighter this term, but she does have some intense classes in biochemistry, physics, etc. At any rate, in researching EBV on the interwebs, I love that word, I found a few things that led to more questions. Apologies if you've covered these before. I did a quick search for EBV in the podcast, but didn't come up with anything. We haven't had an EBV episode, actually. One of the things I read is that children exposed to EBV suffer from milder, inconsequential side effects, runny nose, low fevers, etc. Since the occurrence of the virus is more serious in adolescents and adults, why not expose children to the virus at a young age? Why does the virus cause more serious complications in adolescents and older people? Well, there are some, even in young children, there are some serious complications, so you wouldn't want to just infect them with the wild-type virus. And I'm sure that people have been working on vaccine approaches to this. But I had just talked about EBV this week in lecture, and there are some issues. In particular, you may have immunocompromised kids and you don't know it yet, and they can develop very serious disease. So you don't want to do that. 2, I know EBV is a member of the herpes virus family, and that my daughter will now have this in her system for the rest of her life. Do you have a podcast devoted to this class of viruses and the mechanism of their persistence in the organisms they invade? I know I heard a recent podcast that addressed how the herpes viruses are able to hide out in the body, but any pointers you can provide to podcasts or articles would be appreciated.
Didn't we have Saul on?
Yeah.
Did he talk about EBV?
I don't know if he talked about EBV, but he certainly talked about herpes viruses.
Yeah, that was like number 8 or something. But we haven't done any. I've always wanted to get Elliot Keefe to talk about EBV. But in the meantime, I will put a link to a lecture from this past Monday in my course where I talked about persistence and I talked about EBV. And your daughter will have EBV genomes as episomes in her memory B cells forever. This virus is so clever. It infects B cells and triggers them to become memory cells without having to make antibody. And your memory cells, of course, are long-lived. So that's why you have them forever.
Yeah.
3, I read that young adult exposure to EBV increases the risk of Hodgkin's lymphoma. Has there been any study to explain why this might happen? Does the persistence of EBV in younger children who have been exposed to the virus also include a higher risk for Hodgkin's lymphoma? What about other herpes viruses, e.g. those that cause chickenpox, shingles, cold sores, genital herpes, etc.? Do these include a higher risk for disease or for other diseases? So EBV is basically latent in B cells, and Hodgkin's is if I'm not mistaken, isn't it a tumor of B cells?
I think so.
And so it is related to the persistence of the genome in those cells, and the mechanism is a bit beyond what we have time for right now, but it is Hodgkin's, and Burkitt's lymphoma is another one, another cancer caused by EBV, again by transformation, by immortalization of the cells, and then eventually they can become oncogenic. Herpes simplex viruses, as far as I know, does not lead to cancers. Am I wrong?
Nothing that I can think of.
Can't think of one off the top of my head.
And varicella zoster does not, as far as I know. Cytomegalovirus you haven't mentioned. I don't remember reading that either. All of these viruses, though, are with you forever once you get them. It's absolutely right. 4. I haven't taken your virology course. Look forward to doing so. Feel free to omit this question if it's redundant. I have heard that an immune system challenged by viruses and bacteria is, quote, better, unquote, and made stronger by these repeated challenges. I heard a discussion on a recent podcast, apologies, I did not know which one, where you spoke about the amount of energy required by the body to respond to immune challenges. Is there a cost associated with the generation of a strong immune response and or strong immune system? Does genetics play a part in an individual's immune response? Absolutely, absolutely, and absolutely. Mm-hmm. Genetics play a huge role, and we are now learning that mutations in immune system genes make you susceptible to certain infections. We talked about an energy cost for an immune response, right? Not too long ago. There is a cost, although I don't know how that factors into whether, I mean, that's part of the reason why you don't have a continuous immune response. It has to be dampened.
Right.
And also it would cause damage.
Mm-hmm.
But certainly the education of the immune response is in part done by microbes. I mean, we know that our gut microbiome educates the immune response that is not only local, but is distal to the gut. And we're just learning a lot more about that. There's some evidence that the presence of viruses, at least in a mouse, makes the mouse more resistant to certain bacterial infections. So this is the general idea, and evidence is accumulating slowly.
But we have to be a little careful with this notion that repeated challenges by viruses and bacteria make you stronger, because they'll only do that in the sense of, making your immune system better if they don't hurt you.
Right. And of course, if it's the same virus, you're going to just have a memory response, which
right.
will improve the duration, the durability of memory, I suppose, make more memory cells. But right, as long as
development of effective immune responses makes you stronger and healthier. Immune challenge by viruses and bacteria can have that effect, but it can also, of course, have the effect of making you very sick or dead.
Right. Right. I just thought of another herpes virus that causes tumors, and that's Kaposi's sarcoma virus.
Oh, of course.
Mm-hmm. Can't leave that one out. Otherwise, we'll get letters. We'll probably get letters anyway, but that's fine.
That's fine.
All right. Let's do some picks. Thank you, Anne.
Mm-hmm. Good questions.
Yeah, good
questions.
Very good
Thank
questions.
you. Let's do some picks of the week. Alan, what do you have for us?
I'm just scrolling down to the picks here. What did I end up picking this week? Yes. Heathkit. Do you remember Heathkit?
I used to build Heathkits.
Yeah.
I loved Heath. I built so many of them.
They are coming. They're coming back.
I didn't know they went out of business.
Yeah, they went through a series of changes. People kind of stopped building their kits, partly because people found it easier to buy consumer devices, but largely because Heathkit didn't really update their kits. So, into the 80s, they're still selling stuff with vacuum tubes, and it just didn't really, it was not hip anymore. And so, quite sadly, they went under. I guess the first time they went under was in the early 90s. And they reconfigured themselves into an educational company, and they tried to sell electronics for educational purposes. And then after a little while, that didn't work out. They folded again. And now a bunch of geeks have gotten together, and they haven't disclosed who they are, but it'll be interesting to find out. And they are now restarting the company. They've bought the name, they've bought the website, they had an Ask Me Anything session on Reddit, and talked about a little bit about their plans, and they expect sometime this year to introduce their first kits.
Neat.
I have a Heathkit power supply.
Yeah. Yeah, no, those are common in labs.
Yeah.
Their
stuff, their old stuff, if it was built properly, it often lasts forever. It's huge in ham radio, of course.
Yeah, when I entered Palasi's lab as a PhD student, we needed power supplies. So I ordered 2 Heathkits, and I put them together in a day. And he saw me, and he said, "That's nice, but you should do experiments instead." Okay.
But he probably still has those power supplies. They probably still work.
I bet. It was a lot of fun to build.
Really good investment.
Kathy, what do you have?
Okay, I picked something from a website called Vsauce, and it's distortions. And it's a really cool video. He goes through all kinds of interesting discussion about distortions related to what you might think of sort of as optical illusions, and relates to digital camera lag, something called the rolling shutter effect. And then he launches off into things about astronomy, and points out that, you know, if you looked up in the sky and saw something, I forget what he called it. It looks like some kind of a peace pipe or something, then you'd be hallucinating. But if you looked up something and you saw something that looked kind of like a dipper, then that wouldn't be hallucinating. And he goes through and describes what that would be, and talks about how the constellations, you know, we see everything from our own perspective. But even Voyager 1, which is pretty far out already, isn't far enough away for the constellations to look any different. But if you got far enough away, then, you know, the Big Dipper wouldn't look like a dipper anymore, for example. And I just like the way he presents things. It's very articulate and easy to understand. So cool. I thought it was kind of a cool page.
Yeah, it is very nice. My pick has to do with the fact that I think April is Autism Awareness Month here in the U.S., and there have been a bunch of articles, and I found two of them on Sunday, actually, which I thought were pretty good. One was in the New York Times in the opinion section, and it's by Sam Wang, who's a professor at Princeton. And I always like when scientists, professors write in the Times or newspapers like this, because I think you can learn a lot from it, as opposed to someone interpreting. So he talks about, his article is How to Think About the Risk of Autism. And he says, you know, basically we've had lots of stories on autism, and they still tend to focus on vaccinations and the risks. He said, how do we deal with risk? You know, a study published last week found that the brains of autistic children show abnormalities that are likely to have arisen before birth, yet media coverage focuses on vaccines, which do not cause autism and are given after birth. How can we help people separate real risks from false rumors? So I think it's a pretty nice article about how to do that. And he's got a nice table there of the risks of various things for autism. And the top of the list is if an identical twin is autistic, then the other twin has an 8,300% added risk. Injury to the cerebellum.
At birth.
Prematurity, etc. And the vaccination is the lowest one on the list. Anyway, so it's a good article. I think it's really nicely done.
It is well done. Yeah.
And then the other one is in Science-Based Medicine, which I picked a couple of weeks ago. Various people write for this. This one is by David Gorski. Autism prevalence now estimated to be 1 in 68, and the anti-vaccine movement goes wild. So the CDC just came out with that new figure. 1 in 68 kids born now autistic. It's amazingly high. So he also talks about how to deal with pseudoscience and anti-vaccine stuff and Wakefield, et cetera, and talks about the actual numbers itself and what they mean. So really good articles. And if you're interested in this topic, and I think everyone should read them, it's good. So we had a listener pick up before. Now we have two more here. One is from Marion, who her pick is something called Zome Tool, ZOMETOOL.com. I've known about this for so long, it never occurred to me that this had not been picked. Zome Tool is a toy like Tinker Toys with sticks, struts, and nodes, but the joints have 5, 3, and 2-fold axes of symmetry. It's simple to build structures with icosahedral symmetry, such as a dodecahedron, and icosahedron and a virus, which has both. It was developed by a childhood friend of mine. They sell many kits, for example, Buckyball, DNA, and my favorite, crystallography, which includes a brochure about virus structure written by me. The toys may be used to model many natural structures as well as for beautiful art. So she put some images here. She made
These are cool.
Pretty big ones here.
Yeah.
Yeah,
Huge.
with the mountains in the background. That's way
nice.
cool. Yeah.
I didn't know about these. Anybody know about these before?
No, I didn't.
No.
There you go. That's cool. This is good for classes. You can have people build stuff.
Yeah.
And then Kevin writes, Dear Doctors TWiV, TWIM, and TWIP, I'm a graduate student in the immunology program at Harvard, and I love the TWIX series of podcasts. It's great to hear the perspectives and papers from other fields, especially as I can listen to them on my commute to and from lab or while doing mind-numbing pipetting and tissue culture work. In fact, some fellow graduate students and I were inspired by TWiX to create a podcast about the immune system called Audio Immunity.
Oh.
It's an Alan Dove title.
Wow.
Your audience is our target audience, and anyone that listens will see that we've clearly drawn inspiration from your shows, but we're covering topics from a different angle, and we'll be talking about a lot of topics that you guys don't, though we do have a virology grad student on the payroll. All three of us are also passionate about education, and we're hoping to use the podcast as a tentpole for all kinds of other educational content about the immune system. We've already got some graphics and even a couple of animations that we, actually mostly Matt Woodruff, created that are associated with our episodes. For example, our second episode is about efforts from Michael Nussenzweig's lab to create HIV therapies based on broadly neutralizing antibodies, and we have a graphic of antibodies showing VD and J segments and some animations showing VDJ recombination, as well as my personal favorite, HIV infecting a cell. Right now, we're building these resources associated with the episodes we're doing, but ultimately we're hoping to have a library of graphics, posts, and podcasts that anyone can use to understand the immune system. If you're gracious enough to share our efforts with your audience, everything can be found at immunity.org, and the podcast is also on iTunes. If it's too selfish to ask that my own project be submitted as a listener pick, I thought I'd offer this paper from a few years ago suggesting the use of a phage targeting lactobacilli as a viable strategy to combat beer spoilage. Seems like all the normal objections to using phage as antibiotics in animals, like immune recognition, off-target effects, etc., are irrelevant in this case. That's great. Let's check out that paper for TWIM. Thanks, Kevin, Matt, and Kate.
Yeah, it's great that they're going to have an immunology podcast. Yeah.
So we've had interesting number of podcasts inspired by TWiV, and I have to sometimes make a list of them and see which ones are still extant and so forth. So good luck with that, guys. Remember, getting to number 12 is the hardest. Let's see.
Sticking with something like that is going to be the hard part, especially for grad students. But if you can do it, then do it.
1, 2, 3. All right, so we're on. We have two. We have a beta episode, and then we have episode 0, 1, and 2. So we're up to 4. Keep it up.
Okay.
I haven't listened yet. I actually listened to another podcast, brand new, inspired by TWiV, and I will pick it next time. But I will get to this. I haven't listened, but there's a pick, and go for it, TWiV audience. See what you think.
Yeah.
TWiV is at iTunes, twiv.tv, and if you like us, go over to TWiV on iTunes and give us a rating. That helps to keep us very visible there. And do send us your questions and comments to TWiV at twiv.tv. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was fun. It's good.
And Alan Dove is at turbidplaque.com. You can also find him on Twitter. Thank you, Alan.
Always a pleasure.
You enjoying Twitter?
Yeah, it's okay.
I get notifications when you tweet all the time.
Yeah. I get into conversations occasionally, and it's a good thing to use in moderation.
I told Dixon to do it.
I can quit any time, really.
Yes, of course you can. I told Dixon to do it, but he doesn't want to. He says, I don't know how, and that's his excuse.
But he's on there. He's, isn't he?
No, he's not on Twitter.
He was.
He doesn't do anything, no. It's an imposter, because I know he doesn't do Twitter or Facebook or Google Plus or any.
I thought he had a Twitter account for a little while. Oh, well, anyway.
Nope. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.